Let's take a look at this image from the last video. As you can see, the gene is made up of different basic parts which are colored differently. In synthetic biology, the entire gene is called a composite part or device. Suppose a synthetic biologist is working on our familiar example of a glowing plant. She knows the firefly has a gene coding for a glowing protein. If she adds this entire gene into the DNA of the plant, it will glow too. But she wants to control when it glows. To be specific, she wants it to glow in the dark. As we discussed before, plants have specific promoters whose activity is repressed by light. Such a promoter, combined with the coding sequence for the glowing protein, would do the trick. Well, she's found the coding sequence for the glowing protein from the DNA of the fireflies. She's also found a light-repressible promoter that the plant uses to regulate genes which are only expressed at night. How can she create a device that joins these parts from two different species? Clearly, she needs some way to cut up the respective genes from the plant and firefly, extract the fragments containing the promoter and the coding sequence for the glowing protein, and recombine them into a single device. Fortunately for her, bacteria have evolved a set of enzymes to cut DNA into fragments. They use these to chop up viral DNA if they get infected. These molecular scissors are formally known as restriction enzymes. Here's how they work. First, the enzyme zones in on a particular site or a short sequence of DNA known as a restriction site. Once it comes and binds to that site, it cuts the DNA into two pieces. In our example, the enzyme doesn't cut both strands at the same location, it actually cuts them a few bases apart. This leaves each side of the cut with one extended strand, forming an overhang. Each restriction enzyme binds to a different binding site and therefore creates different overhangs. Alright, this shows us how to cut two genes. Let's say our synthetic biologist has looked through both genes, the plant gene containing the promoter and the firefly gene with the coding sequence for the glowing protein. And wow, she's lucky! She's found restriction sites for her favorite enzyme on both genes, and she's been able to cut out the gene fragment she needs. Now, notice that they were cut using the same enzyme, they have the same overhangs. This means that they have complementary DNA bases, which allows the overhangs to stick to each other. An enzyme called a DNA ligase can be used to stitch the small gaps that remain on either strand. And voila, we have a device made of both these parts. But is this likely? Let's look at how chancy the whole scenario is. We have to use the same enzyme to cut both genes to get complementary overhangs. While we can cut the two genes using different enzymes, we can't stick them back together afterwards. We have to have the same restriction site on both genes. As you realize, the odds are not in her favor. Even if they were, any device she puts together would be done using the restriction enzyme she's acquired. Other scientists might use different restriction enzymes to cut the same genes. The overhangs on the part cut by different enzymes are going to be different as they target different sites. Synthetic biology would be, in fact it used to be, a complete mess. So how did the SynBio community solve this problem? The answer is staring us in the face. Use the same restriction enzymes everywhere. Every part that a synthetic biologist uses typically goes through a series of molecular modifications to insert standard restriction sites both upstream and downstream of the part so that they can be easily combined. Let's see this with the example of the BioBrick standard. A SynBio part, like this coding sequence for the glowing protein, can be made into a BioBrick by adding specific restriction sites upstream and downstream of the part. These are called the prefix and suffix sequences respectively. These restriction sites also don't occur anywhere inside the part's own sequence. Modifying a part to have this prefix and suffix is a long and complicated process, and we won't dive into that today. By adding two BioBrick parts together, along with the right enzymes, we can cut them, pair them up, and stitch them together to create a device. And you know what's cool? Adding together those two BioBricks in this way just leaves us with a BioBrick. The device as a whole follows the same set of rules as the parts, as you can see. One thing we must emphasize, this video is filled with examples, not general rules. There's a huge variety of restriction enzymes. each cutting DNA at different sites and resulting in differently shaped cuts. Each kind of cut has its own requirements and its own standard procedure. The point of this video is not biobricks. It's that to put parts together as a synthetic biologist requires some modification, some standard rules that make them compatible. This is really the same as building something with Lego blocks. Lego blocks are made according to precise measurements so that the pegs and holes fit perfectly. Standardization is just the process of deciding what those precise constraints should be.